Aqeedah, who will be presenting work on childhood trauma and genetic variation in the DAT 40 base pair VNTR contribute to HIV-associated neurocognitive disorders.
Thank you. Let me just get my slides up.
Yeah, we can see them and hear you.
Perfect, thank you. Hi everyone, I'm Aqeedah. I'm a first-year master's student in Stellenbosch University's Neuropsychiatric Genetics Research Group, and the research I'm presenting today takes a look at the environmental and genetic factors contributing towards HAND. HIV-associated neurocognitive disorders, or HAND, refers to cognitive impairment, emotional disturbances, and motor abnormalities that occur as a result of HIV-induced neuroinflammation and neurotransmitter dysregulation. The diagnosis of HAND relies on a number of neurocognitive tests, which assesses functioning on several cognitive domains. Although these tests provide a diagnosis of HAND, other methods or markers of disease would be beneficial in understanding HAND in countries with a high HIV prevalence, such as South Africa. There is evidence of both environmental and biological factors contributing towards the progression of HAND. The combined effects of environmental stresses, such as childhood trauma, has been associated with reduced verbal fluency and executive functioning. Through shared underlying neuroinflammatory mechanisms, environmental stresses and HAND may overlap in symptomology, resulting in a heightened combined effect on behavior and cognition. When it comes to biological factors, the neurodegenerative effects of HIV are attributed to the migration of active HIV-infected monocytes across the blood-brain barrier and into the brain, leading to neuroinflammation. These inflammatory factors lead to neuronal injury and cell death and result in synaptic and dendritic loss within the brain of HIV patients, and this damage may contribute to the neurocognitive decline experienced by HAND patients. Dopamine is a neurotransmitter that is strongly associated with the reward systems of the brain and plays a role in modulating learning and motivation. Cognitive functioning depends on this dopaminergic signaling within the prefrontal cortex and striatum of the brain. An intermediate level of dopamine is optimal, with altered levels resulting in a decrease in cognitive functioning, as seen in HIV-associated dementia. HIV infection has also been shown to result in an increase in extracellular dopamine, as well as accelerated neuropathology. Studies have provided valuable evidence of the neurocognitive effects of altered dopamine metabolism, transport, and functioning as a result of genetic variation. And so for this project, we looked at two genes. The first is the COMT gene, which encodes catechol-O-methyltransferase, and this plays a role in the metabolism of dopamine in the prefrontal cortex, an area linked to cognition, executive functioning, and memory. The homozygous MET genotype of the COMT Val158 MET polymorphism has been associated with increased dopamine signaling in the prefrontal cortex. The other gene that we looked at is the DAT gene, which encodes the dopamine transporter, and this is a key protein necessary for the uptake of dopamine, with dysfunctional DAT leading to depleted intracellular levels of dopamine. Here, the homozygous 10-repeat allele of the DAT40 base pair's variable number of tandem repeats has been associated with increased cerebrospinal fluid dopamine in HIV patients. For our research, 113 participants were recruited by researchers in the Department of Psychiatry for a larger study. The recruitment of participants, the assessment of childhood trauma, as well as neurocognitive assessment was conducted by trained personnel at baseline and then again at a one-year follow-up. Blood samples were drawn from participants from which DNA was extracted for genetic analysis. I then used this DNA to conduct a polymerase chain reaction for the COMT and DAT variants of interest. The COMT amplicons then underwent an additional step of restriction enzyme digest before both COMT and DAT products could be genotyped on acrylamide and agarose gels respectively. Sanger sequencing was used to validate the genotype obtained on the gels. Analysis of variants was used to examine the effect of including predictive variable interactions on the model fit. HIV seropositivity was associated with a reduction in cognitive scores at follow-up, and we also noted a trend level HIV and childhood trauma interaction effect on cognitive scores. For the sake of time, I'm going to focus on the results related to the genetic analysis. So when considering genotype, our first finding showed that the combination of HIV and the DAT 10-repeat homozygosity was associated with reduced global cognitive scores in longitudinal models. And we also found that including the interaction between the DAT-10-10 repeat, childhood trauma, and HIV explained significantly more of the variance in longitudinal cognitive scores. We found no significant common genotype associations. Moving forward, studies can be done to determine whether interaction between COMT and DAT are involved in the development of hand or how other neurotransmitter-related genes may be associated. Of course, this would only be possible with a much larger sample size, which would also aid in determining more accurate statistical measurements. Another limitation was that the childhood trauma questionnaire used is reliant on self-report measures of trauma, which may be influenced by recall bias, and the questionnaire does not distinguish between single and repeated traumatic events. And so we hope that future research would be able to adequately address these limitations. At present, the study provides some evidence of the combined effect of genetic and environmental influences on cognition, which may prove beneficial in identifying individuals at a greater risk for developing hand. Insight into the pathways and mechanisms that govern these interactions will facilitate a better global understanding of the effect on the long-term cognitive functioning. Thank you.
Thank you so much for your presentation. It was great. Do we have any questions from the audience? While we wait for the audience, I have a question. I saw your study was on females only. Do you think that these results would be similar in males?
I would think so, based on international studies. So international studies have looked at males and females, and as far as I know, there are no sex differences in terms of the effect of the genes on dopamine variation, but I'm not sure about HIV though.
Okay.
Or childhood trauma. What I can say is that in South Africa especially, women are at a higher risk of experiencing childhood trauma, and so that might also affect the outcome.
Yeah, that makes a lot of sense. Are there any other questions from the audience? Here we go. Great presentation and work. Are there variables like socioeconomic status or education level of parents, etc., that could influence the outcome?
I think when doing this type of study, it's important to, with the tests that we use, like the neurocognitive type of test, it's important to standardize those things because their performance on certain tests might fluctuate based on socioeconomic status. So I don't think that genetically, I don't think that like the outcome of the test would differ, but I think it's important to standardize that.
Great, thank you. Are there any other questions from the audience? If not, we can move on to the next presenter. Thank you, Aqeedah, for your great presentation.